All
Thank
right,
you.
so as I mentioned in the presentation, the previous presentation, we used ExPASy, which is has been for quite a number of years now the portal for SIB resources, and we had this glycomics tab created to not only put our own stuff but also advertise what other people are doing. And I'm going to give you a few examples of what we have. Unfortunately, we are a little bit in a transition for some useful tools that I cannot demonstrate because they are broken. So you better know they're broken. The substructure search is broken and the epitope extractor is broken. And we are in the process of reshaping all of this so that it will be integrated again into GlyConnect. So basically, I'm focusing on GlyConnect, which integrates a lot of the tools that we are developing. But first, I would like to just mention that we're trying to change the look of ExPASy, which is a bit stiff with database on the one hand and tools on the other, and doesn't say much. Although each time you can you can click on more and have a description, but that doesn't tell you everything you need to know. So we're trying to do this a bit more interactively and have it like you can zoom in. So here you have portals where I have the glycoscience.de I mentioned before, which was the ancestors of all the portals. So glygen, glycosmos rings is the former, is also the ancestor of glycosmos. So I don't know if I should keep it. Kyoko will tell me whether, and because then
it's actually separate.
Well, it has no. I mean, it has common information, but it's
rings is a sorry. Go ahead, but it's for tools, and glycosmos is for data. So that's kind of how I yeah.
Okay, so this is what I put data, so I should data mining, so algorithmic and data mining tools to aid biology research. Whereas I put for glycosmos, whoops, sorry, repositories and data. So if you can correct me, but I think I got the text from ExPASy, so since we co-wrote it, it should be okay. So the idea is to have different categories, and in each category have, so here we have 2D, 3D, Cazymes. So here, for instance, you have database like CASI or Cake Pathway, Kyoko's glycan pathway predictor, a small tool for digesting, simulating digestion, the oglycolog here, which is also a simulation tool for pathways, and we collaborate. This is developed by Andrew McDonald and Gavin Davey in Ireland, and we're collaborating with them. So we are trying to have a bit more of an interactive map showing the different, so the mass spectrometry resources, some utilities. On the structure here, you have glycan and CSDB, which is the database hosted in Russia regarding bacterial, archaeal, plant, and fungal carbohydrate. So this is towards which we're going in terms of presentation, to show the different categories and maybe have a little bit more idea of what goes with what. So this is under construction. Now I would like to start with a UniProt entry, because we have the links. If you go to the PTM processing, this is usually where you have the annotation regarding glycosylation sites. So here is a good entry with lots of publication supporting the evidence of a glycosylation site here. Nonetheless, a lot of these of these publications are like large-scale proteomic papers where you have glycosylation site identified but no glycan identified there. So to see a bit further, you can actually click on the glyconnect link, and you have here the description in glyconnect that is giving you a different picture on the on this protein, where you see actually the taxonomy, that's the protein. So you see the sources. So in very much the same way, Kyoko has been putting forward the ontology that she refers to. So we try to do the same. So for the anatomy, we're using the Uberon ontology. For when we have no Uberon ontology term, we use a Go term, and we also use the Cellosaurus, and so the cell line ontology provided by the Cellosaurus when it applies. So you can see here that this is the summary for this particular protein. We have 10 defined structures. We have 61 compositions. It's involved in 4 diseases. We have 13 papers that describe the information that is there. There are 6 sites that are reported, and also some structures not attached to a site because it's not provided. And when we have mass spectrometry experiments, we have the peptides on which the site was actually identified. So these different papers, the 13 papers, they spread so here there's one paper describing this site. So the information that this structure is on these sites. And so you can find this information on this paper. So usually when you have a very detailed, so I'll probably, we have, you see here that I have a very detailed structure with all the linkages that are provided. So this is from a 1993 paper, but then the site is undefined. So it's, as I said, sometimes you have to choose between very precise structure and no information, site-specific information. So what we do is that we use the site-specific experiment to suggest some sites. So this structure is potentially on site 70 or 270, 271 due to the site-specific experiments that are there. And we can find the structure here. No, here I think was the one. No, that's the fucosylated one. So I need the, I think, without the fucose. Sorry, I made a mistake. But I could take the fucose one, which is here, which is also undefined. There's a suggested site due to the site here, 70, the suggested structure matching. So we're trying to match these very unspecific structures, but specific site experiments with those very specific structures with very poor information on the site, which is due to the papers here that are a bit old, and the technique wasn't there to do all the mapping. So this is the information you find in a protein page, and you have also the view here with the plugin to see the SNFG glycans when they're there and all the possible view. So this is the light mole. This is developed by David Senal in Czech Republic. And you can see this is now in the PDBE. This is the default viewer in the PDBE version, European version of the PDB. So this is the summary for a protein. And so here, if you see, for instance, if you want to see one of the diseases, this protein is involved in a disease. So you can see that in that particular disease, same here we have ontology, disease ontology that is describing the disease. And you have all the sugars that are on which site, so which protein and which site they are found. So we can actually go, whoops, to the one of the protein here in the, what did I do? Yes, this protein is involved in the disease, so I selected it. And one view you can have of the information is provided by Compositor, which is, the paper is under revision at the moment. We are about to publish it. And Compositor actually is trying to help you see the possible relationship between those different structures. Obviously, some of them are substructures of the others. And instead of looking very deeply into the pathway, the enzymatic pathways that are synthesizing these structures, we propose a very broad representation where you have these compositions that are related by just a change of one monosaccharide added. So if you see here, this is exactly what is associated with the macroglobulin here, where you have lots of compositions, and it's sort of hard to see what is the content. But with this tool, you can see it's all tied together, except for a few exceptions here that are left alone and not part of the network. But in fact, all of these compositions are linked to one another. And you can see that you can go from this composition to this one by adding a few co's. Here, you go from this composition to this one by adding sialic acid. Here you go from this composition to this one with a, here you just add an exose. So you can see it's a mesh of composition tied together by probably the action of a glycosyltransferase that adds one monosaccharide. So you can have a snapshot of what is sialylated, what is fucosylated, what is not either sialylated, I mean neutral, so no sialic acid. What are the high mannose structures in the network? The size of the node is actually the number of papers, the number of references that back the existence of that composition. And the one in the middle is only showing that there's one possible structure that is associated with this protein, which is a bit more defined than a composition. So we have 3 defined structures here that you can see, and all the rest is composition. And so you see that in the graph. You can actually explore this and see what is the consistency. And you see here this virtual node, which is here in gray. And it means that if we didn't have this node, then these 2 composition would be isolated from the rest. And so we allow to, so when there's only one step difference in terms of adding a monosaccharide, we're allowed to insert a virtual node. Here, this is a very, you can see you have 12 hexos and 2 hexnac, so it's a really large structure, and there's nothing in the network here that is close enough in one step to get to such a large structure. So this is the way to visualize the information differently. And I actually selected one of the leftovers composition, like this one, and you can see it's not connected. So you can actually also investigate a little bit more. You can see we are always cross-linking to glycan for each of our information. And this 491F1 that we have here, we can actually look at the sequences. So if I look at composition here, we can look at the sequences in a range of 30 amino acids to see. So I have to remember again, I said 491. So 4 exos, 9 XNAC, 1 fucose, and linked. And I can have a look at the alignment. Demo effect, fantastic. Did I do the right one? 491. So I thought I did it this morning, and of course it doesn't work now. So anyway, you see an alignment. Maybe I should have, let me just check for one thing here. Composition, go 491 and fine. So, okay, I'll just go to the next tool, but you should see if I have still in my, no, I don't, sorry. You should see an alignment of the sites that is depending on the composition, and you can see that you have some conserved amino acid that seems to depend on the composition, especially if you have a fucose or not a fucose, has a big bearing on the alignment. So the other tool to visualize, so what we are very well aware of is that our data is biased because we collect papers. So people are interested in they're more interested in human than in any other organism. They're more interested in N glycans than they are in O glycans. So there's always, if we input the data in GlyConnect, there's unfortunately a bias that is imposed by the bias introduced in experimental data that are out there. So we propose the tools to actually see the data under different lights so that you are not too constrained by the bias and you can have a look at what is out there. So if I take this GlyConnect octopus, which actually offers the possibility of selecting some properties of amino acids, of glycans. So here, I've mentioned that before, fucosylated, glycosylated, silylated. So here, imagine I have already pre-prepared that so that there wouldn't be any demo effect. So my composition here was tetraentenary. It was fucosylated. So I've actually, and it was not silylated. There was no sialic acid here. So you can actually see that I have tetraentenary, non-silylated, co-fucosylated. And then I can see in the database what corresponds to this particular type of information. So I can link composition with, I can see with the number of proteins. So this is again in Homo sapiens. I have a number of tissues in which it's expressed. So it's actually noted here. And you can see how populated some tissues are as opposed to others. You can see how populated some, how popular, actually I should say, some compositions are with respect to some protein, and you can actually see whatever you want. If you want to see the structures rather than the composition, so you put the structures here, and then you see the structures, and they are numbered, so it's not particularly easy to see, but if you mouse over the name, you can always see the structure. And all the same, you can see the connectivity of the data, how this is all linked together or not. And so this is offering an idea of how much data do I have regarding these different entities. So I have here used GlyConnect to compare, for instance, the content in glycan of serotransferrin versus lactose transferrin. So you can actually see that there's some overlap. So you have basically 60 glycans for each, and 50% overlaps, and the other 50% is different. So you have the distribution here, and you can see it's a little bit different. And what I have done is to export the everything that is common here. I can only keep what is common to the two, copy it here, and I can go to this. I've done it so that it doesn't take time. I've pasted all of that in the custom tab of the GlyConnect, and we have our 32 common link, and I can see in the database how popular these common structures are. So here in all of GlyConnect, attached to this composition, I have all of these structures. So 31 for this one, 9 for this one. I need some virtual node to connect the whole thing. And I can sort of have a look in more detail at maybe the specific composition as opposed to the very common composition. So this one is a very common high mannose, apparently 30 possible structures. And so I can navigate from one view to the other and ask myself question on how relevant would be, or how consistent would be a set of composition, possibly to identify the glycans, and especially in the framework of mass spectrometry analysis, where you need a composition file as an input parameter. How can I shape my input file with this exploration that is possible around protein, but also around tissues, around cell line, around disease, and so on. So we developed lately that tool to explore the data in GlyConnect. I think I will stop here.
Thank you very much. Thank you, Frédéric.
No problem.
We can have, again, questions, a little time for questions. You can use the chat for that, or you can unmute and use the microphone. There's a comment from Casper. Really nice multi-armored octopus.
Yes, now it's multi-armed indeed, with all sorts of possibilities. So you could see in the, maybe you could not, but in the octopus, this is dev. So it's under development. It's not quite finished. We had a version before in GlyConnect that was only limited to having composition in the middle, proteins on the one hand, and tissue or structures we could change. But now you can really customize it exactly the way you want to see the different information, and you can here also query by determinants or by cores. So everything is made much more possible for this one in the new version.